Hey y'all, what's up? So coming to you on Friday, right? After protein folding day. All right. So as if you remember, these sections represent our amino acids. These amino acids, right, are monomers of a protein. So these have to come together. These are the pieces that build the protein. These are the amino acids we get through either diet or our body can make most of them, but this is what protein comes from. Not pipe cleaners, but amino acid. So the first thing that happens in our primary structure is these amino acids have to bond together, and this bond is called a peptide bond. So I have amino A and amino B, right? We're going to bond those together, right? So now we got these bonded together. I'm going to come on and go and bond another one on here, right? Like so. Okay, so now we have a peptide, right? A couple of these bonding together, we get a peptide. I'm going to keep on rolling. I'm going to bond another one on here, right? So as this gets longer, as this peptide gets longer, right, from a couple amino acids to many amino acids, we have a poly meaning many, we have a polypeptide chain. And the order that these come together is not random, right? So I'm kind of randomly putting them together right now, but it's not random, right? It is our genetic material, our DNA has the key that tells us the order to put these things in depending on the structure we want to build. And the structure we build is going to determine the function of what we want, right? So, there's tens of thousands of different structures of proteins in our bodies. So there's tens of thousands of different functions for them as well. So this is our primary structure, a polypeptide chain. It's bonded together by a peptide bond. And each one of these is an amino acid, our monomers. So now moving on to our secondary structure. You have the alpha helix. So a helix is like a helix staircase, right? It's like a winding staircase shape, right? So we can kind of get that going, something like this right here. Okay, that didn't work out. Pencil. All right, so wrap the sucker around this pencil like that. Oh, it broke. That ain't good. All right, so gonna have our alpha helix shape there, right? We can kind of make our beta plated sheet here, which will be something along the lines of this right here. So now we got like our beta plated sheet. So our alpha helix, our beta plated sheets. So this is kind of our secondary structure, right? So now moving on to the tertiary structure, several different things can happen. We start getting a lot of different intermolecular forces. R groups, which we'll learn about later, start to be attracting or repelling. There might be some sulfide bridges. There might be some hydrogen bonding, some polar interactions. But so what's going to happen? So say these two greens come together and form a disulfide bridge, right? So now we get that. That kind of hooks on there. So right, but maybe these two are polar opposite or have the same charge. Maybe they push apart on each other. And then maybe these two are oppositely charged, so they attract together, right? So, well, that brings these two closer together, right? And maybe we get a hydrogen bond in there and it pulls that there. And then maybe this non-polar object gets pushed down into the middle, and so forth and so on, all these forces till you end up with like a globular shape here, right? Now, it could end up more fibrous, like long and kind of cylindrical. I said that word right. I'm pretty sure that's impressive. So this will be things for like your hair, right? Long cylindrical things or whatnot. But so you end up with like this. This is our tertiary structure. So now for our quaternary, right? Quarter without the R, right? Quaternary structure is when you take two tertiary structures or more, two or more tertiary structures and combine them, now we would have a functioning protein in the quaternary structure. So, just walk you through the protein folding. Hope this kind of helped clear up any issues. I'm going home. It's the weekend, baby. See y'all Monday.